Welcome back, Life Sciences. I hope you had a good but a quick break. Get that oxygen flowing, get the blood flowing. It clears the mind and helps us to study and remember better. We are recapping the cell today, right? We're looking at the concepts. What is a cell? How does transport occur in the cell? And all of these functions, right? These organelles that we're going to look at that we find in the cell that have a particular function. We ended off last time with looking at the concept of what the cell membrane was. Now, if we have a look again at the structure of the cell membrane controls what is going to move in and what is going to move out. That's a really important concept when we're looking at this whole selectively permeable. I know another word is differentially permeable that you often use, but as I said, I can spell that better, and that's why I use it. Now, the cell membrane is going to control what moves in and out of the cell. Now, when it comes to transport in the cell, things need to move. And as I've said before, how do we get things moving? We're going to speak about a concentration gradient. And what we're talking about, a high concentration means a lot of something. And a low concentration means less. Now, as I said to you before, imagine this is a hill, okay? What would be easier for me? To get things up the hill or to get things down the hill? And the easiest is to go from a high to a low. And your body feels exactly the same way. Why must I spend energy on doing something that's going to be energy expenditure when I can do things the easier way? Remember, it's not so much about the easiest way, but it's what is efficient. So I've got to move things around my body all of the time. Plants have got to move things around their structures all of the time. The easiest we can do it, the less energy we can use, that's going to be far more efficient. So how do I move things in and out, right, of a plant or an animal? The first thing I'm going to look at is diffusion. And simply what diffusion is moving a substance. Here's my high concentration. If I look over here, this is going to be my low concentration. So what the molecules want to do is they're going to go from a high to a low, and they are going to spread equally. Okay, I'm going to show you a concept, right, that you're going to come across later. So if, for example, here I'm going to draw, this is a leaf. Okay, and at the bottom of the leaf, there are little holes that can open and close. They are called stomata. And what happens is a plant needs to photosynthesize, and the plant needs carbon dioxide. So what happens is the plant will always bring carbon dioxide, will move from a high concentration out of the plant into a low concentration in the plant. Now, I know this is a weird concept for you, but what has it been able to do? That process of diffusion, moving something from a high to a low, actually enables, right, the plant to photosynthesize. Exactly the same happens to us. In our lungs, all right, when we breathe in, oxygen comes into our body and moves from a high concentration via diffusion, because it's a gas, to a low concentration in our blood. And the carbon dioxide moves from a high in our blood to a low, all right, in our lungs. That's how we breathe. That's how we can bring air in. We can bring in nice fresh air, bring out. So all these parts have it, how we're going to move things around the body. So diffusion is one part. The next concept, all right, is osmosis. Now we follow the same, all right, the same concept where we're going to move water. Right, now this is really important. What moves? Water must move from a high concentration to a low concentration. Now, there is a bit of a terms and condition here, because when water moves, it must move through a semi-permeable membrane. If I've got a little jug over here and I put water in here and the particles just move around, that is going to be diffusion. But if you have a look over here, what I need to do when osmosis occurs, okay, is that I need to have, all right, a concentration gradient. Now, if you have a look, this pink is the water. These little blue things are what's dissolved in the water, okay? So say, for example, it could be sugar, it could be salt, and they are dissolved in the water. So I've got more water this side and less water because I've got more dissolved substances. So this has a high water potential. So where does my water move? From an area of high concentration to an area of low concentration. Have a look at the level. The level is going to change, because if my water moves from one area to another, then so is my level of water going to change. Now, guys, this is really important that you understand. When you look at questions like this, and we're going to do one now, you need to look at this. Where is there the most water, and where is there the most dissolved substances? So as I said to you, transport, osmosis, high to low, but through a semi-permeable membrane. Okay, so here is a possible question that we can ask you relating to the process of osmosis. So here we have a grade 10 learners. They set up an apparatus below, right, in the school lab using whisking tubing. Now, you might not have any idea, or you've never heard anything called whisking tubing before, but in brackets, they've explained what it is. They've said that whisking tubing allows water to move in and out like the cell membrane of the cell. So what is it telling you here? It's telling you that this membrane is selectively permeable. I know, as I said, differentially, some of you might even use the word semi-permeable. So let's have a look at the setup over here. So I've got a beaker, all right, and what I've done is I filled the beaker with pure water. So I'm going to change my pen color quickly. This, guys, is filled, all right, with pure water. So I'm going to write here, it has a high water potential. Now what I've done is, if I'm going to change my colors back, I have now put a semi-permeable membrane, this little whisking tubule, and inside here I have made a strong sugar solution, okay? So here I've got this concentration gradient. I've got water on the outside, and inside, in this little sack, I've got the strong sugar solution. So what question are they going to ask me? Okay, so they're still carrying on with how they set it up. The learners recorded the level of the water in the beaker before the start of the investigation. Right, so that's telling me that there is the water level is going to be important. Next thing they said, after 2 hours, there was a change in the level of water in the beaker. So here is my water level. Guys, 2 things could have happened to it only. The water level could have gone up, or the water level could have gone down. Now, the question is, state the process that is investigated in the diagram below. Now, let's have a look. I had water on the outside. I had sugar on the inside. And in between the two of them, I had a semi-permeable membrane. So obviously the process that I was going to look at was osmosis. Okay, there we go. I had water, I had sugar, and I had this membrane in between them. Now I have to explain, all right, tell me why the changes that were observed in the level of the strong sugar solution in the visqueen tubing and the pure water in the glass beaker. Okay, guys, two things they want. Let's break it down. So, what did you notice about the sugar solution, and what did you notice about the water in the glass beaker? Now, osmosis, if we know osmosis occurred, what happened is, guys, where does water move? So let's start. Water moved from an area of high water potential, that means lots of water, in the beaker to, all right, the tubing. So what happened here? Water moved from the beaker into the tubing, which means, let's answer it, all right, the level of the tubing increased. There we answer the second one. Okay, then that's the next thing. So as the pure water moved out of the beaker, the level decreased. Okay, so guys, what happened? This level got lower and this got more. Because where does water move? From a high concentration to a low concentration. Okay, so those are kind of questions that you're going to get. Have a look. Do you have a concentration gradient? Do I have a high water potential? Do I have a really strong concentration of sugar or salt? That is usually the idea that you're going to have when you're going to do an osmosis experiment. So just keep that in mind when you do that. As I said to you before, just make sure when you get these that you're able to explain the process of osmosis.